Hello biology lovers, are you ready to unveil the new topic or the next topic of our sexual reproduction of humans, that is the gametogenesis? Let's dive into spermatogenesis and oogenesis. Welcome to cell to system. Now let's break the term gametogenesis. Gametogenesis is formed by two terms, that is gametes and genesis. Gametes means the specialized haploid cells for reproduction. In case of males, the gamete is sperm, and in case of females, the gamete is ovum. Now, genesis means creation. So, the gametogenesis clearly means creation of gametes. The gametogenesis in humans is of two types. One is spermatogenesis and another one is oogenesis. Testes, and more precisely said, the seminiferous tubules of the testes is the location where the spermatogenesis occurs. Before knowing about spermatogenesis, we need to understand the anatomical structure of a testis. The almond-shaped structure in case of male, the main gonads, is differentiated into external coverings and also internal structures. There are three coverings which are present outside of the testes. The first one is tunica vaginalis. The another one is tunica albuginea. The innermost is tunica vasculosa. The tunica vaginalis is the outermost layer. The tunica albuginea is the middle layer. It is the fibrous tissue layer. The tunica vasculosa is the innermost covering of the testes. Through the LS of the testes, we can precisely see the three layers. The outermost layer is the tunica vaginalis, which protects all the internal parts of the testes. The next layer, tunica albuginea. It is the tough fibrous capsule beneath the tunica vaginalis that forms inward septa, dividing the testes into almost 250 testicular lobes. The tunica vasculosa, it is the innermost layer and has been provided with the vascular supply. Now coming to the internal structures, the first internal structure is the testicular lobules. The second one is the seminiferous tubules. The third one is interstitial tissue. And the fourth one is retitestes and duct system. So starting with testicular lobules, as I have already said, this tunica albuginea, this differentiates the inward of the testes into about 250 testicular lobules. These lobules, the main function of the testes occurs here, that is the spermatogenesis is occurring inside the testicular lobules. Each testicular lobules contains 1 to 3 coiled seminiferous tubule contains 1 to 3 seminiferous tubules. In seminiferous tubules, the spermatogenesis occurs. And also there is a nutritive cell called Sertoli cell, which provides nutrition to the developing sperm. These Sertoli cells are also known to secrete an enzyme called inhibin. The another tissue that is interstitial tissue or interstitial cells, which is the place between two seminiferous tubules. In this place, the Leydig cells occur. We have already discussed the function of Leydig cells. So you let me know in the comment section what is the function of Leydig cell. Coming to the retitestis, each seminiferous tubule ends into a network of ducts or network of tubes. These are known as retitestis. These retitestis ends into a duct called vasa efferentia. The transverse section of testes shows this diagram. In this diagram, we can clearly see the seminiferous tubules in which the spermatogenesis occurs. The primary spermatocytes are very precisely seen. The nutritive cells here are the Sertoli cells. Sertoli cells are elongated cells which also produces an enzyme called inhibin that inhibits the function of luteinizing hormone and follicle stimulating hormone. Some blood vessels are also seen. In the center of the lobules, we can see the mature sperms. Here, the interstitial cells produces the Leydig cells, in which the testosterone is also produced. Seminiferous tubules open into a network of channels called retitestis. The retitestis pass into the vasa efferentia, another channel of network. This way, the sperm, when formed into seminiferous tubules, goes to the retitestis, then to the vasa efferentia and epididymis, then vas deferens, and after passing through the vas deferens, it mixes up with the fluids of accessory gland and forms the semen. Semen is then transferred to ejaculatory duct, to the penis, and finally to the outside of the body. If the fertilization occurs, the semen is transferred to the female reproductive tract. The process of spermatogenesis occurs in the seminiferous tubules of the testes. The seminiferous tubules are located inside the testicular lobules. The duration of spermatogenesis is 64 to 74 days in case of humans. This is the duration, and it requires a lot of ATP. 64 to 74 days. This process of spermatogenesis is supported by two cells. One is Leydig cell, which is responsible for the production of testosterone, under the influence of luteinizing hormone secreted from the anterior pituitary gland. Next one is Sertoli cells, which is the nutritive cells for developing sperms. Now coming to the main process. Spermatogenesis process, the spermatogonium or the germ layer cells, they undergo further repeated mitotic divisions and produce two types of cells which form a group. The type A cells, they are the cells which finally grows into a sperm or which finally becomes a sperm, and the type B cells, they are the reserved cells for the germ layer. If I draw the structure, so if it is the spermatogonium and it is diploid in number, so this will undergo mitosis to form type B and type A cells. Type A cells will continue with the spermatogenesis process. It will grow into primary spermatocyte, which is again diploid, and here mitosis occurs. It is also diploid. The primary spermatocyte undergoes meiosis 1 to form secondary spermatocyte. The ploidy of secondary spermatocyte is haploid because here the meiosis 1, the reductional division occurs. The ploidy is haploid in nature. This secondary spermatocyte, they undergo meiosis 2 and form haploid spermatids. These spermatids goes various morphological changes to grow into a mature sperm. So here you can see each primary spermatocyte forms 4 spermatids and finally the sperms. Here we can differentiate the spermatogenesis process into 3 phases. The first one, which is the formation of or the multiplication of spermatogonium, is known as multiplication. Multiplication phase. The next one, that is primary spermatocyte growing into secondary spermatocyte, it is known as growth phase. And finally the spermatid. In the growth phase, this occurs. Lastly, the most important phase of spermatogenesis is the spermiogenesis, where these spermatids, they finally grow into the mature motile sperms. These are the 3 phases of spermatogenesis: multiplication phase, growth phase, and the spermiogenesis. Here, this is the full phase of spermatogenesis. Now, you need to understand what are the key changes occurring here so that this structure is morphologically changed into this. Now, let's understand. During the process of spermiogenesis, the spermatid, it changes into this mature sperm. So let's understand the structures the mature sperm has. Acrosome, it is a enzyme-rich structure which is present at the head of the sperm. Acrosome helps to dissolve the walls of the ovum during fertilization. The whole part is the head of the sperm. This is the plasma membrane present in the head. This is the neck portion which is very flexible in nature, and the tail, it is actually the flagellum. Now coming to the changes occurring. The formation of acrosome is a very unique change occurring in the sperm of the human beings. This acrosome possesses Golgi apparatus. The tail is the flagellum. The nucleus of the sperm is condensed to form a very reduce the structure. The excess cytoplasm is removed to provide the sperm with a sleek structure to move into the female reproductive tract very precisely and in a very good way. This is all about spermatogenesis process, and also we have studied about the ultrastructure of the testes. In the next class, we will be knowing about the ovary and the oogenesis process. Till time that, please stay tuned with Cell2 system. Don't forget to subscribe my channel and press the bell icon. Thank you so much.